Very good evening, one and all. Thanks for joining us for this national level technical workshop on basic PCR protocol and mitochondrial DNA based nested PCR for identification of cell lines. So, I would just like to say that I'm waiting for some more time for the candidates to join. It's almost 5 p.m. Welcome, Prabha. Yes, Vikas, Abhishek, Netra, Sarabjit, Dr. Vivek, Seema, okay, Usha, Siddharth, Priyanka, Krishna, okay, Sudharshini, yeah, very good evening. So we are also live on YouTube and Facebook. Okay. So it's almost 5:05. So let me start, and I'm going to accept the candidates one by one. If someone is joining in between the workshop. So today what topics I'm going to cover is what is the background behind PCR, what is PCR, different modifications in PCR, as well as what are the cycling conditions in PCR, what components and reagents we have to take in PCR, and how to document your PCR product and its various applications. A very good evening, guys. Yes, very good evening. So let me introduce myself. This is SHRADHA Mahendra. I am a research scholar at Department of Genetics and Biotechnology, OSMANIA University, Hyderabad. My specialization is cancer biology. I'm the joint director and also co-founder of Novagen Biologicals, life science-based startup, where we are working in different areas of life sciences. Our broad focus is biotechnology, where we are working for the research and developmental activities in the area of life sciences. At present, we are working on microalgae applications for human and animal welfare, as well as our interest is in recombinant proteins, where we are working on CHO cell lines. So let me start today with basic PCR protocol, which is a day one technical workshop. So I would like to talk about the background behind this polymerase chain reaction. So as we all know that in microbiology, microscope is the latest invention where we can view the microorganisms under the microscope, which we cannot view our naked eye. Whereas compared to molecular biology, PCR is the greatest invention, and at present, recently, CRISPR-Cas9 genome editing technology, it has also been invented. So the development of the polymerase chain reaction is one of those innovations that changed the course of molecular science with its impact spanning countless subdisciplines in biology. So the theoretical process was first outlined by Kepe and co-workers in 1971. However, it was another 14 years until the complete PCR procedure it was described, and it was experimentally applied by Carey-Mullis while at Cetus Corporation in 1985. Automation and refinement of this technique, it progressed with the introduction of a thermal stable DNA polymerase from the bacterium Thermus aquaticus. Consequently, we called it as Taq DNA polymerase or Taq-Pol. So one of the most powerful and revolutionary procedures which are used today to analyze the DNA sequence of our interest is based simply on the unique biochemistry of DNA replication, and it is called as polymerase chain reaction. So this powerful technique, it allows the researchers to amplify specific sequence of DNA from samples that contain only a few individual DNA molecules to sequence complementary to the primers. Most PCR methods, they typically amplify DNA fragments of up to 10 kilobase pairs, although some techniques allow for amplification of fragments up to 40 kb in size. However, PCR methods is not without problems. As all the technologies come with some pros and cons, PCR method also comes with certain pros and cons. So the extreme sensitivity of PCR increases the probability that false priming or DNA contamination, it will lead to the amplification of the wrong molecule. One way to help prevent these artifacts that arises from the impurities is to work in an extremely controlled environment and to purify the reagents and primers prior to use. So what you have to do is you have to work in extremely controlled environment or the closed environments away from dust particles, and you need to purify your reagents and primers before you go for this PCR. So after the completion of PCR reaction, the buffer, which is a mixture of primers and free nucleotides, and now the newly synthesized DNA fragments, it need to be separated so that the synthesized DNA molecule, it can be used for further downstream applications. So now what is PCR? What is the definition of PCR? PCR is a powerful amplification and a versatile technique for copying DNA that can generate an ample supply of a specific segment of DNA, that is an amplicon, from only a small amount of starting material. So for carrying this polymerase chain reaction, that is PCR, we need not need large amount of samples, only small amount of sample is needed, which we call as DNA template or target sequence. PCR allows a single DNA sequence to be copied millions of times or altered in predetermined ways. So while straightforward and generally it is trouble-free, there are pitfalls that complicate the reaction, producing the spurious results. When PCR fails, it can lead to many non-specific DNA products of varying sizes that appear as a ladder or smear of bands on agarose gel electrophoresis. Sometimes you get no product at all. So another potential problem which occurs is when mutations are unintentionally introduced in the amplicons, resulting in a heterogeneous population of PCR products. So now let us see what are the various modifications of polymerase chain reaction. So the first one is allele specific PCR. This is a diagnostic or a cloning technique which is used to identify or utilize SNPs, that is single nucleotide polymorphisms or single base difference in DNA. It requires prior knowledge of a DNA sequence, including differences between alleles, and uses primers whose 3' ends encompass the SNP. So for carrying this allele specific PCR, you need to have a proper knowledge of your DNA sequence of interest. So next one is assembly PCR. So it is the artificial synthesis of long DNA sequences by performing PCR on a pool of long nucleotides with short overlapping segments. As the name suggests, assembly PCR, it is causing the pool of long nucleotides, oligonucleotides with short overlapping segments. That's why it is called as assembly PCR. Now what is asymmetric PCR? It is used to preferentially amplify one strand of the original DNA more than the other. That's why it is called as asymmetric PCR, because it is causing one strand to amplify properly when compared to the original DNA more than the other. What is colony PCR? So bacterial colonies like E. coli, it can rapidly be screened by PCR for correct DNA vector constructs. So for performing correct DNA vector constructs, you need to perform this colony PCR. Now what is helicase dependent amplification? So this technique is similar to a traditional PCR, but it uses a constant temperature rather than cycling through denaturation, annealing and extension. So in conventional traditional PCR, what we are doing, we are performing it through different temperatures like denaturation, annealing and extension. But in case of helicase dependent amplification, we are taking only a constant temperature rather than cycling through denaturation, annealing and extension cycles. Now what is hot start PCR? This is a technique that reduces the non-specific amplification during the initial setup stages of PCR. So this technique is commonly used in research laboratories because we don't want to have any non-specific amplification, because we want to have only a specific amplification of our DNA product or a PCR product. So what is inter-sequence specific PCR? A PCR method for DNA fingerprinting that amplifies regions between some simple sequence repeat. So it is amplifying the regions between the simple sequence repeat to produce a unique fingerprint of amplified fragment lengths. That's why it is called as inter-sequence specific PCR. Now let us see what is inverse PCR. So it is a method which is used to allow the PCR when only one internal sequence is known. This is especially useful in identifying the flanking sequences to various genomic inserts. What is ligation mediated PCR? So this method, it uses small DNA linkers which are ligated to the DNA of interest and multiple primers annealing to the DNA linkers. It has been used for DNA sequencing, genome walking and DNA footprinting. So that's why it is called as ligation mediated PCR. Now let us see what is methylation specific PCR. It was developed by Stephen Balin and Jim Hubban at the Johns Hopkins School of Medicine, and it is used to detect the methylation of CpG islands in genomic DNA. This methylation specific PCR, it uses the quantitative PCR, can also be performed to obtain the quantity rather than the quality information about your methylation. So that's why it is called as a methylation specific PCR, where you are using a quantitative PCR which gives you the quantity of your methylation rather than the quality information. Now what is multiplex ligation dependent probe amplification? It permits the multiple targets to be amplified with only a single pair of primers. Here you can use only a single pair of primers, where you can amplify your multiple targets with only a single primer pair. Now what is multiplex PCR? As the name suggests, it uses the multiple primers, unique primer sets within a single PCR reaction to produce the amplicons of varying sizes specific to different DNA sequences. That's why it is called as multiplex PCR. Now what is nested PCR? This PCR is also very much used in research labs because it increases the specificity of DNA amplification by reducing the background due to non-specific amplification of DNA. Now, nested PCR is often more successful in specifically amplifying long DNA fragments than the conventional PCR, but it requires the more detailed knowledge of target sequence. To carry this nested PCR, you need to have a detailed knowledge of your target sequences. Now, what is overlap extension PCR? It is a genetic engineering technique which allows the construction of a DNA sequence with an alteration which is inserted beyond the limit of the longest practical primer length. Now let us see what is quantitative PCR. It is used to measure the quantity of a PCR product, preferably in real time. That's why it is called as quantitative PCR. It is a method of choice to quantitatively measure the starting amounts of DNA, cDNA, that is complementary DNA, or RNA. Quantitative PCR is commonly used to determine whether a DNA sequence is present in a sample and also the number of its copies in the sample. So the method with currently the highest level of accuracy is quantitative real-time PCR. So in this PCR, we are knowing whether a DNA sequence is present as well as the number of copies in the sample. Now what is RT-PCR, which is also called as reverse transcriptase PCR? Many people get confused between quantitative PCR and real-time PCR, but it has little bit difference between these two PCRs. It is a method used to amplify, isolate or identify a known sequence from a cellular or tissue RNA. Real-time PCR is widely used in expression profile. So this is the main difference between quantitative PCR and real-time PCR. In real-time PCR, you can also perform your gene expression studies to determine the gene expression or to identify the sequence of an RNA transcript, including transcription start and termination sites, and if the genomic DNA sequence of a gene is known. So to map the location of exons and introns in the gene as well. Now what is tail PCR, which is called as thermal asymmetric intercalated PCR? It is used to isolate the unknown sequence flanking a known sequence. Within the known sequence, this tail PCR is using a nested pair of primers with different annealing temperatures. So here what tail PCR is doing, it uses a nested PCR pair of primers with differing annealing temperatures. A degenerate primer is used to amplify in other direction from unknown sequence. Now what is touchdown PCR? This PCR also used in research labs very frequently. A variant of PCR that aims to reduce the non-specific background by gradually lowering the annealing temperatures as PCR cycling progresses. Now what is gradient PCR? Gradient PCR is a technique that allows the empirical determination of an optimal annealing temperature using the least number of steps. This optimization can often be achieved in one experiment. The master cycler gradient provides a gradient function that in one single run it evaluates up to 12 different annealing, elongation or denaturation temperatures. So in gradient PCR you can keep different temperatures in a single run. During the same run, a number of possible concentration parameters can also be tested row by row. So what is ARMS PCR? So the allele-specific PCR, which is also called as ARMS PCR, that is Amplification Refractory Mutation System, or PCR Amplification of Specific Allele, or ASPCR, which is used to detect the single nucleotide polymorphisms. So the allele-specific PCR is also called as Amplification Refractory Mutation System because it uses two different primers for two different alleles. Refractory means it is resistant to something. Now two sets of primers are designed in this PCR. The mutant set of the primer is refractory, that is resistant to the normal PCR, and the normal set of primers are refractory to the mutant PCR reaction. That is why it is called as Amplification Refractory Mutation System. Now what are the components and reagents which are used in this polymerase chain reaction? We need DNA template that contains the DNA region or target to be amplified, which is very important. Primers, which is a short synthetic oligonucleotide which is used in many molecular techniques from PCR to DNA sequencing, which is complementary to DNA region at the 5' 3' ends of DNA. These primers are designed to have a sequence which is the reverse complement of a region of template or target DNA to which we wish primers to anneal. So the primers should be complementary to your DNA target so that it can get annealed to your DNA template. So designing primers. So designing appropriate primers is very essential to the successful outcome of a PCR experiment. When designing a set of primers to a specific region of DNA which is desired for amplification, one primer should get annealed to the plus strand, which by convention is oriented in the 5' 3' direction, which we also called as sense or a non-template strand. So remember, 5' 3' direction is also known as a sense or a non-template strand. And the other primer, it should complement the minus strand, which is oriented in the 3' 5' direction, that is antisense or a template strand. So there are also a few common problems that arises when you design a primer. So what are those problems? So self-annealing of primers resulting in formation of secondary structures such as hairpin loops. Primer, they anneal to each other rather than binding to the DNA template, creating primer dimers. Drastically different melting temperatures for each primer, making it difficult to select an annealing temperature that will allow both the primers to efficiently bind to target sequence during thermal cycling. So what considerations we have to take care while designing of primers? So the primer length, it should be short, like 15 to 30 nucleotide residues or the bases. So our GC content, it should range between 40 to 60%. Very important consideration while designing primer. The 3' end of primer should contain a G or a C in order to clamp the primer and prevent the breathing of ends, increasing the priming efficiency. So now what is this breathing of ends? So DNA breathing occurs when ends they do not stay annealed but they fray or they split apart. The three hydrogen bonds in GC pairs, it helps prevent the breathing, but it also increases the melting temperature of the primers. So the 3' ends of a primer set, which includes a plus strand primer and a minus strand primer, it should be complementary to each other, nor can the 3' end of a single primer be complementary to other sequences in the primer. These two scenarios is responsible for primer dimers and the hairpin loop structures respectively. So optimum melting temperatures for primers, it should range between 52 to 58 degrees Celsius, although the range can be expanded to 45 to 65 degrees Celsius. So the final TM for both the primers, it should differ by no more than 5 degrees centigrade. So this is very important consideration when you are designing a primer, that the melting temperature, it should not be more than, the difference should not be more than 5 degrees Celsius. Dinucleotide repeats, for example, GC, GC, GC or AT, AT or single base runs like A or C, 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 it should be avoided as they can cause the slipping along the prime segment of DNA and they can cause the hairpin loop structures to form. If unavoidable due to nature of DNA template, then only include the repeats or the single base pair runs with a maximum of 4 bases. So there are many computer-aided programs in designing the primer pairs. What you can do is you can go to this NCBI primer designing tool, that is National Center for Biotechnology Information, which is mostly recommended for designing of a primers, and also Primer 3 tool, which will be helpful for designing your primers. So in order to avoid the amplification of related pseudogenes or homologs, what you can do is before designing primers, you can run a blast on NCBI, which is nothing but basic local alignment search tool to check the target specificity of your primers. So another important component for carrying this PCR is DNA polymerase, such as Taq polymerase at around 70 degrees Celsius temperature, because it works around 70 to 80 degrees Celsius temperature. That's why we are using Taq DNA polymerase. Deoxynucleoside triphosphate, that is DNTPs. So these are the building blocks from which the DNA polymerase synthesizes a new DNA strand. Buffer solution. It provides a suitable chemical environment for optimum activity of DNA polymerase and also the stability. Divalent cations. So magnesium or manganese ions, generally MgCl2 is used, but manganese can be utilized for PCR mediated DNA mutagenesis, because as the higher concentration of manganese concentration, it increases the error rate during your DNA synthesis. That's why we want to use magnesium chloride instead of manganese. So next is template DNA. So what is this template DNA? So the DNA fragment to be amplified, it should not be greater than about 3 kb in length, and ideally it should be less than 1 kb fragment. But fragments up to 10 kb, it can be amplified by standard PCR technique, but longer the fragment, less efficient will be your amplification. So the integrity is more important than the amount of your DNA. So you have to take into consideration that you need the integrity of your DNA should be more when compared to the amount of your DNA. So linear DNA is amplified more efficiently than the circular DNA. So plasmid DNA, it should be digested beforehand. What is DNA polymerase? So this DNA polymerase is an enzyme that assists in DNA replication. Such enzymes, they catalyzes the polymerization of deoxyribonucleotides alongside a DNA strand, which they read and use as a template. So now the newly polymerized molecule is complementary to the template strand and it is identical to the template partner strand. So this DNA polymerase is considered to be a holoenzyme since it requires a magnesium ion as a cofactor to function properly. In the absence of magnesium ion, it is referred to as apoenzyme. So the earliest PCR experiment, they utilized the clinofragments of Escherichia coli DNA polymerase 1 at a temperature of 37 degrees Celsius to amplify the specific targets from human genomic DNA. What is Taq polymerase? polymerase is a thermostable DNA polymerase named after the thermophilic bacterium, that is Thermus aquaticus, from which it was originally isolated. It is abbreviated as TACpol and it is frequently used in PCR, a method for amplifying short segments of DNA. So Thermus aquaticus is a bacterium that lives in hot springs and hydrothermal vents, and Taq polymerase, it was identified as an enzyme which is able to withstand the protein denaturing conditions, that is high temperatures, which is required during your PCR. Therefore, this is the main reason that it replaced the DNA polymerase from Escherichia coli, originally which is used in PCR. That's why instead of DNA polymerase from E. coli, we are using Taq polymerase because it can withstand the high temperatures. Taq's temperature optimum for activity is 75 to 80 degrees Celsius with a half-life of 9 minutes at 97.5 degrees Celsius, and it can replicate 1,000 base pair strands of DNA in less than 10 seconds at 72 degrees Celsius. So one of the Taq's drawback is its relatively low replication fidelity. So it lacks a 3' to 5' exonuclease proofreading activity, and it has an error rate which is measured at about 1 in 90,000 nucleotides. So this is one of the main drawbacks of using Taq polymerase. Now let us see what are the different cycling conditions. So first is initialization step. So this step consists of heating the reaction to a temperature of 94 to 96 degrees Celsius, which is held for about 1 to 9 minutes. What is denaturation step? This step is the first regular cycling event and consists of heating the reaction to 94 to 98 degrees centigrade for 20 to 30 seconds. So before denaturation step, we want to carry the initialization step, which is the heating the reaction to a temperature of 94 to 96 degrees Celsius, which we want to hold at 1 to 9 minutes. After that only we are going to carry with our first step, that is the denaturation step, at a temperature of 94 to 98 degrees Celsius for about 20 to 30 seconds. So it causes the melting of DNA template and primers by disrupting the hydrogen bonds between complementary bases of DNA strands, yielding single strands of DNA. So in denaturation step, our double-stranded DNA is splitting into single-stranded DNA because of the denaturation. Now what is annealing step? So this is the next step in PCR. The reaction temperature is lowered to 50 to 65 degrees Celsius for 20 to 40 seconds, which allows the annealing of primers to the single-stranded DNA template. Typically, the annealing temperature is about 3 to 5 degrees Celsius below the Tm of the primers used. Stable DNA-DNA hydrogen bonds are only formed when the primer sequence very closely, it matches the template sequence. The polymerase binds to the primer template hybrid and it begins the DNA synthesis. So in annealing step, our primers are coming and binding to our DNA sequence of interest when we lower the temperature, because at high temperature our primers cannot anneal. Now the next step is extension or the elongation step. So the temperature at this step, it depends on the DNA polymerase used. Taq polymerase has its optimum activity at a temperature around 75 to 80 degrees centigrade, and commonly a temperature at around 72 degrees Celsius is used with this enzyme. So at this step, what is happening is the DNA polymerase, it synthesizes a new DNA strand which is complementary to the DNA template strand by adding the DNTPs that are complementary to the template in 5' to 3' direction. So in elongation step, our DNTPs are getting added for the DNA extension. So the extension time, it depends both on the DNA polymerase used and on the length of DNA fragments to be amplified. So now the next step is the final elongation step. So this step is occasionally performed at a temperature of 72 to 74 degrees centigrade for about 5 to 15 minutes after the last PCR cycle, that is after the extension step, to ensure that any remaining single-stranded DNA is fully extended. So in this step, we want to confirm and we want to be ensure that any of the left single stranded DNA is fully extended. So we want to carry this final elongation. Now final hold. This step we carry out at 4 to 15 degrees centigrade for an indefinite time. It may be employed for short-term storage of the reaction. So in the final hold step, all your extension step is completed and final elongation is completed, and then only we are carrying this final hold at a temperature of 4 to 15 degrees centigrade. We want to keep it at a 4 degrees centigrade, and we can carry out this step of final hold. So we don't want any of the PCR product to get amplified, and we want to stop the non-specific amplification of secondary products. So that's why we carry this final hold step. Now, as I said you, these are different cycling conditions. So this is standard 3-step PCR cycling. Initial denaturation, which we are carrying at a temperature of 94 to 98 degrees centigrade for 1 minute. As I said you, 1 to 9 minutes. So number of cycles, only 1 cycles. So denaturation at a temperature of 94 degrees centigrade for almost 10 to 60 seconds, that is 30 seconds. And annealing and extension 5 degrees centigrade below the TM, that is at 70 to 80 degrees centigrade, your extension step, which we carry for 25 to 35 cycles. So total polymerase chain reaction cycling conditions we are going to carry for 25 to 35 cycles. So final extension we are carrying at 70 to 80 degrees centigrade for 5 minutes, and that too for 1 cycles, that is number of cycles is 1 cycles. And then hold for 4 degrees centigrade for indefinite time, and that is also for 1 cycle. So we are carrying 1 cycles. So almost for PCR cycling we need to perform 25 to 35 cycles. Even you can perform more than 35 cycles, but it will give you non-specific or the secondary products which you don't require, because you require your product, concerned product. Now what are the materials and reagents which are used? So when setting up a PCR experiment, it is important to be prepared. Wear gloves to avoid contaminating the reaction mixture or reagents. You need to include a negative control and if possible a positive control when you carry this PCR. Arrange all the reagents which are needed for PCR experiment in a freshly filled ice bucket and let them thaw completely before you set up this PCR reaction, because you don't want any of the nuclease activity and degradation. That's why you need to carry all your PCR steps in ice filled bucket and let all your reagents and chemicals thaw completely before you make a master mix of your PCR. Keep the reagents on ice throughout the experiment. Standard PCR reagents include a set of appropriate primers for the desired target gene or DNA segment to be amplified, DNA polymerase, buffer for specific DNA polymerase, DNTPs, DNA template, and sterile water or double distilled water. Additional reagents may include magnesium salts. If magnesium salt is not included in your buffer, you can add it separately also. Materials include PCR tubes and caps, a PCR tube rack, an ethanol resistant marker, and a set of micropipetters that dispense between 1 to 10 microliters, 2 to 20, 20 to 200, and 200 to 1000 microliters, as well as a thermocycler. So when setting up several PCR experiments that all uses the same reagents apart from your DNA template, they can be scaled together and combined together in a master mix. So you can carry the master mix in a sterile 1.8 or 2 ml of microcentrifuge tube. So to analyze the amplicons resulting from PCR experiment, reagents and equipment for agarose gel electrophoresis is required. So to approximate the size of a PCR product, an approximate commercially available molecular weight size standard is also needed, because you want to detect your PCR product, the band size of your PCR product, with the molecular weight size standard marker. So when you compare with the molecular weight size standard marker, which have different base pairs, you can know that your product is 50 base pairs or 109 base pairs with molecular weight size standard marker only. So PCR reagents in the order they should be added. So these are the reagents like sterile water, PCR buffer, DNTPs, MgCl2, your forward and reverse primer, your template DNA and tag DNA polymerase. So first you need to add your buffer, then DNTP. You need to add, I'm just showing you this for 50 microliter of reaction. So you need to add a 5 microliter, that is 1x concentration of your PCR buffer. So this 5x concentration or a 10x concentration of PCR buffer, first you have to make it to 1x concentration, and then this 1x concentration you have to take 5 microliter from this 1x concentration. TNTPs you need to add 1 microliter, MgCl2 mostly 3 microliter, your forward and reverse primer 1 microliter each, and your template DNA, it is variable. You can add even 0.5 microliter or 1 microliter depending on your quantification of DNA, your quality of DNA. And tag polymerase, we want to take 5 units per microliter, that is 0.5 microliter. And then when you last add your water, sterile distilled water or a double distilled water, you need to calculate that how much your 5 microliter buffer, 1 microliter of your DNTPs, 3 microliter of MgCl2, your forward reverse primer, that is 1 microliter each, and your template DNA and your tag polymerase. After calculation, rest you have to add the water. And then you have to make up the volume to 50 microliter. So now let us see what is basic PCR protocol. So you need to place a 96 well plate into the ice bucket as a holder for 0.2 ml thin walled PCR tubes. So the PCR tubes are very small, that is they are 0.2 to 0.5 ml. It allows the PCR reagents to be added into the cold 0.2 ml thin walled PCR tubes. It will help prevent nucleus activity and non-specific priming. So this is the main reason that you have to carry your PCR reaction into your cold ice chill bucket to prevent the nucleus activity and non-specific priming, as I have already mentioned. Pipette the following PCR reagents in the following order into a 0.2 ml thin walled PCR tube: sterile water, 10x PCR buffer, DNTPs, MgCl2, primers, that is both forward and reverse primers, and your template DNA. Since experiment should have at least a negative control and possibly a positive control, so it is recommended to set up your master mix in a 1.8 ml or a 2 ml of microcentrifuge tube. So in a separate 0.2 ml thin walled PCR tubes, you need to add all the reagents with the exception of template DNA. So you need to keep into consideration that for a negative control, you need not add a template DNA, because when you add a template DNA, it will become a positive control. So increase the water to compensate for the missing volume, but you need not add a template DNA for your negative control. In addition, another reaction, if reagents are available when you do a master mix, should contain a positive control using template DNA and or primers which are previously known to amplify under the same conditions as the experimental PCR tubes. So a negative control is devoid of your DNA template, but in a positive control, you are using a DNA template. So this is the difference between a negative control and a positive control. Tag DNA polymerase is typically stored in a 50% of glycerol solution, and for complete dispersal in the reaction mix, it requires a gentle mixing of your PCR reagents by pipetting up and down at least 20 times. So now you need to add all the reagents and chemicals, and you need to do pipetting, proper pipetting, so all the reagents should mix properly. At least you need to mix it for 20 times. So the micropipetter, it should be set to about half the reaction volume of the master mix when you are mixing all your master mix and the reagents, and care should be taken to avoid introducing the bubbles. So this is the main reason that you want to keep your micropipetter to half the reaction volume, because if your reaction volume is 50 microliter, just set your micropipetter to 25 microliter and mix the master mix properly and without any air bubbles. Avoid using air bubbles. Put caps on the 0.2 ml thin walled PCR tubes and place them into thermal cycler. So after adding everything and your template DNA at last, what you need to do is properly you need to cap this small 0.2 ml wall tubes, because if you don't put your caps properly, that when you are running your PCR cycle or a thermal cycler, your caps can open in between the reaction, and then all your product will be exhausted and you cannot see the amplification when you run on agarose gel electrophoresis. So you need to carry it very very patiently and slowly. So once the lid to the thermal cycler is firmly closed, you need to start the program. So when the program has finished, the 0.2 ml thin walled PCR tubes, it may be removed and stored at 4 degrees Celsius. So the PCR products, it can be detected by loading the aliquots of each reaction into the wells of agarose gel, then stain the DNA that has migrated into the gel following the electrophoresis with the ethidium bromide. So after carrying your PCR, which will run for almost 2 to 2.5 hours, you need to stop the program. You need to take out your 0.2 ml PCR tubes, which contains your template DNA and your final PCR products. And before running into agarose gel electrophoresis, you can keep your PCR tubes at 4 degrees centigrade, and you can go for preparing your agarose gel electrophoresis by adding the ethidium bromide after preparing the agarose gel solution. If a PCR product is present, the ethidium bromide, it is a carcinogen, and it is also a very good dye which will intercalate between the bases of the DNA strands, which allow the bands to be visualized with a UV illuminator. So this is the reason we want to use ethidium bromide, because it will intercalate between the bases of your DNA strands, but it is also carcinogen. So now the total number of copies of double-stranded DNA, it can be calculated by using the following equation. So number of copies of DNA is equal to your DNA amount in nanograms into 6.022 into 10 to the power of 23 divided by length of DNA into 1 into 10 to the power of 9 nanogram per ml into 650 daltons. Now calculate the number of copies of DNA is used to determine how much template is needed per reaction. So using this formula, you can know that how much template you need to take per reaction. False positives may occur as a consequence of carryover from another PCR reaction, which would be visualized as multiple undesired products on an agarose gel electrophoresis. Therefore, it is very important to use a proper technique, including a negative control and a positive control when it is possible. So, how you will detect false positive means, when you perform your PCR and when you are performing the new PCR, PCR tends to show some of the false positive results in your current PCR from the carryover from your another PCR reaction. And you can visualize this only when you keep a negative control. When you load a negative control in your agarose gel electrophoresis, when it will show a band in a negative control, it means that you are carrying the false positives from your previous reaction. So this is the main reason we want to keep negative control along with the positive control to check if our reaction is contaminated or not. So while ethidium bromide is the most common stain for nucleic acids, there are several safe and less toxic alternatives also. The following are the alternatives, including methylene blue, crystal violet, cyber safe and gel red. So even you can use these dyes compared to ethidium bromide, which is a carcinogen, but mostly labs are using ethidium bromide. Now, how to calculate a melting temperature for your primer? So knowing the melting temperature of the primers is very imperative for the successful PCR experiment. However, nearest neighboring thermodynamic models are preferred over the more conventional calculation, that is TM 4 GC, that is guanine cytosine, plus 2 AT, that is adenine and thymine. The former will give more accurate TM. So when you do calculate the melting temperature with your thermodynamic model, it will give you very accurate value when compared to when you are calculating with 4 into GC plus 2 AT. So the TM estimation, it happens because it takes into account the stacking energy of neighboring base pairs. So this thermodynamic model is taking into account the stacking energy of your neighboring base pairs, which gives you a very accurate TM estimation when compared to the later one. But you can do very simple calculation with this later one. That's why you are not using, most of the people are not using this thermodynamic models. Now, how to set up a thermal cycling conditions? PCR thermal cyclers rapidly heat and cool the reaction mixture, which allow for the heat-induced denaturation of your duplex DNA, that is strand separation, annealing of primers to the plus and minus strand of DNA template, that is single-stranded DNA, which has been denaturated in denaturation step, and elongation of the PCR product. Cycling times are calculated based on the size of template and your GC content, that is guanine cytosine content of the DNA. So the general formula starts with an initial denaturation step at 94 degrees centigrade to 98 degrees centigrade, depending on the optimum temperature for DNA polymerase activity and GC content of the template DNA. So a typical reaction, it will start with a 1 minute denaturation at 94. So any longer than 3 minutes, it may inactivate your DNA polymerase, which destroy its enzymatic activity. So one method known as hot start PCR, it drastically extends the initial denaturation time from 3 minutes to 9 minutes. So when you do this hot start PCR, what it is doing is, it is extending the initial denaturation time from 1 minute or 3 minutes to 9 minutes. With hot start PCR, this DNA polymerase is added after the initial exaggerated denaturation step is finished. So we want to stop this denaturation. So that's why what we are doing is, we are adding the DNA polymerase after the initial exaggerated denaturation step is finished. So this protocol modification avoids the likely inactivation of your DNA polymerase enzyme. The next step is to set the thermal cycler to initiate the first of 25 to 35 rounds of your 3-step temperature cycle. So as I told you that we are going to carry 25 to 35 cycles of PCR. While increasing the number of cycles above 35, it will result in a greater quantity of PCR products, but too many rounds often results in the enrichment of undesirable secondary product. As I already said you, when you carry more than 35 cycles, you will get more PCR products, no doubt. But what it is causing is, too many rounds, it results in the enrichment of undesirable secondary products as well. So the 3 temperature setup steps in a single cycle, it accomplishes 3 important tasks. The first step, it denatures your template DNA. The second step, it allows the optimal annealing of primers. And third step, it permits the DNA polymerase to bind to the DNA template and synthesize the PCR product. Now the duration and temperature of each step within a cycle, it may be altered to optimize the production of desired amplicon. Now the time for the denaturation step is kept as short as possible, usually 10 to 60 seconds. It is sufficient for most of the DNA templates. So the denaturation time and temperature, it may vary depending on your GC content of the template DNA, as well as the ramp rate, which is the time it takes the thermal cycler to change from one temperature to the next temperature. So the temperature for this step is usually the same as that which is used for the initial denaturation phase, that is, it is 94 degrees centigrade. Now a 30-second annealing step follows within the cycle at a temperature which you set about 5 degrees Celsius below your apparent TM of the primers, ideally between 52 to 58 degrees centigrade. So the cycle concludes with an elongation step. The temperature depends on DNA polymerase selected for the experiment. For example, the stacked DNA polymerase, it has an optimum elongation temperature of 70 to 80 degrees Celsius, and it requires a 1 minute time to elongate the first 2 kb. Then it requires an extra minute for each additional 1 kb to be amplified. So PFU DNA polymerase is also another thermostable enzyme that has an optimum elongation temperature of 75 degrees centigrade. So most of the researchers also use this PFU DNA polymerase instead of tag DNA polymerase, because this also having a temperature of 70 to almost 80 degrees centigrade, or you can also say 75 degrees centigrade. PFUDA polymerase is recommended for use in PCR and primary extension reactions that requires the high fidelity, and it requires 2 minutes for every 1 kV to be amplified. So the final phase of thermal cycling, it incorporates an extended elongation period of 5 minutes or longer. So this last step, it allows the synthesis of many uncomplicated amplicons to finish, and in case of tag DNA polymerase, it permits the addition of an adenine residue to the 3' ends of all the PCR products. So as I have said that we are carrying this final phase of thermal cycling to see that any of the single stranded DNA which is not extended properly in the extension step, we want to do it the final phase of thermal cycling to elongate, to extend it further. And this is the last step which will allow the synthesis of many of the uncomplicated amplicons. So any of the single stranded DNA which is remaining in your extension or the elongation, in final phase what is happening is the uncomplicated amplicons or the single stranded DNA further get extended. In the case of tag DNA polymerase, it also permits the addition of adenine residues to the 3' ends of all the PCR products. So this modification is mediated by terminal transferase activity of tag DNA polymerase and is useful for subsequent molecular cloning procedures that require a 3' overhang. So that is the reason adenine residue we are adding, or we are doing an addition of an adenine residue to our 3' end of PCR products for subsequent molecular cloning procedures that require a 3' overhang. Termination of the reaction is achieved by chilling the mixture to 4 degrees Celsius and also by addition of EDTA. By this you can also terminate reaction by addition of EDTA as well. So now let us see how to carry this agarose gel electrophoresis after completion of PCR product and after storing your 0.2 ml of PCR tubes at 4 degrees centigrade. We want to go for agarose gel electrophoresis. Now before loading our PCR products, the final PCR products, and to know the band size and to know is amplification has really happened, our primers are proper or not, and our reaction is not contaminated, we want to run this agarose gel electrophoresis. So agarose gel electrophoresis, it is a powerful separation method frequently used to analyze the DNA fragments generated by restriction enzymes, and it is a convenient analytical method for separating DNA fragments of varying sizes ranging from 100 base pairs to 25 kb. So DNA fragments which are smaller than 100 base pairs are more efficiently separated using polyacrylamide gel electrophoresis. Now, what is the protocol behind this agarose gel electrophoresis for separation of your DNA fragments or your PCR products? Reagents and materials for preparation. We need a tank, tray, comb, normal melting point agarose powder, 10x TV buffer solution, gel stain, that is we need almost EcoSafe nucleic acid staining solution. We are using ethidium bromide, microwave oven, flask, measuring cylinder. And for loading of your PCR product, we need pipettes, PCR tubes or the tin foil, power supply. For documentation, we need a camera, gel documentation system, and also we need UV illuminator, trans illuminator, in which we need to see our gel with our PCR product. First, you need to prepare agarose gel. So for a 2% agarose gel, you need to measure 2 grams of agarose in an Erlenmeyer flask and add 100 ml of 1x, or even you can add a 50 ml of your 1x TBE buffer. Now cover the flask with a parafilm and heat in the microwave until your agarose dissolves. So leave it to cool down about 60 degrees Celsius, or you can cool this agarose solution at a room temperature on bench for several minutes. But you need not do it for a long time because it will solidify. Then you cannot pour this agarose gel in a gel tray. So you need to do it for several minutes, but do not leave it too long. So the agarose should not start to solidify before loading into a gel tray. Stain the agarose solution with 5 microliters of your ethidium bromide. After adding ethidium bromide, mix the agarose solution well by swirling the flask. Pour the agarose which you have made into a mold that is 3 to 5 mm thickness in your casting tray. After 30 minutes at room temperature, carefully remove the comb. So after pouring your agarose gel into your gel casting tray, you need to keep a comb on your agarose gel, and then you need to wait for 30 minutes so that your gel will get properly solidified, and then slowly you have to remove the combs, and you will have different wells with your agarose gel in your agarose gels. Position the gel into the gel electrophoresis tank. So after this gets solidified, your agarose gel gets solidified in a gel tray, you need to take out your agarose and you need to place this into an electrophoretic tank, and you need to avoid bubbles. Add enough TB buffer to cover the gel to a depth of about 5 mm. So the gel which you are keeping in an electrophoretic or a gel electrophoretic tank, it has to be added with a proper TB buffer to cover the gel to a depth of 5 mm. We are adding a TB buffer so that your DNA it will properly travel, with the buffer solution in the buffer solution. Now, how to do the loading? So mix the DNA samples with gel loading buffer. So you cannot just take your DNA samples of PCR product and you cannot load into the well. Without your gel loading buffer, you cannot load your DNA samples because it won't be visible when you view it under UV illuminator. So mix your DNA samples with gel loading buffer with pipettes properly. You need to take a 5 microliter of your buffer plus DNA solution, that is your PCR product. So about 0.3 to 0.5 micrograms of DNA, it gives a visible band of your 1/10 of PCR reaction volume. Prepare marker DNA of known sizes, that is 6 microliter from Promega marker. Even you can take a standard ready-made molecular marker to know your PCR product size. And also you can prepare a DNA of your own size, like known size. What is loading dye consists of? I am showing you the loading dye of 6S concentration, that is 0.25% of your bromophenol blue. You can take 25 mg of your bromophenol blue and 0.25% of xylene xylone. You can also take it 25 mg plus 30% of glycerol in water. Now you have to store at 40 degree Celsius. Now you have to mix all this loading dye properly, and now this loading dye you have to take, this become your buffer solution, and this loading dye you have to take with your DNA solution, and then only you have to load your DNA samples into the gel, into the wells. Load the mixture slowly into the slots without any air bubbles. Avoid making any air bubbles. Attach the electrical leads so that DNA can move towards the anode, because our DNA is negatively charged, so it has to move towards the anode that is positively charged, that is red lead. So you need to apply a voltage of 1 to 5 voltage per centimeter. Now run the gel until the gel loading buffer stain has migrated the appropriate distance, that is normally until your bromophenol blue dye front it migrated 3/4 of the way down the gel. And in the last well, you need to load your molecular weight marker also, because you need to know that what is your product size. So it is very important to load the molecular standard molecular weight marker of different DNA band sizes, because you need to compare it with your PCR product, that what is the size of your PCR product. So it is very important, and even you can load a negative control as well and positive control. Now let us come to documentation. Then after the gel has run, so now load the mixture slowly into the slots. Avoid making bubbles. Now attach the electrical leads after you properly load your samples and your molecular weight marker, so the DNA can move from cathode to anode, which is a red lead, which is a positively charged, because your DNA is highly negative charged, so it has to move from your black cathode to your anode, that is red lead. You need to apply a voltage of 1 to 5 voltage per centimeter, and now you need to connect the electrodes, switch on the power, and now let the gel to run until the gel loading buffer stain has migrated the appropriate distance of 3 to 4 of the way down the gel. After that, you can switch off your gel electrophoretic tank, and you can take out your gel, but you need to wear gloves while taking out the gel, because you have added the ethidium bromide into the agarose gel, which is highly carcinogenic, and anything without apron and without gloves you need not handle. So let us see the documentation part. After taking out the gel, you need to check it. Turn off the current and remove the leads. Examine the gels. Carefully place it on an ultraviolet trans illuminator and take a photo. So this is the gel picture. On the right hand side, you can see a ladder of a standard molecular weight marker of different base pairs, that is 100 base pairs, 500, 1 kb and 3 kb. And on the left hand side, you can see different sizes of your product, that is 765 base pairs, 880 base pairs, 1022 base pairs, 765 base pairs. So now you can compare with your standard molecular weight markers your different base pairs, which is clearly visible in this gel picture. You can take a picture, or you can also save this picture if your trans illuminator is connected to your system, and you can save it. Now let us see what are the applications of PCR. Why we are using PCR? PCR can be used to amplify specific genes which is present in different individuals. PCR products can be used for cloning and sequencing to obtain information on the mutational changes in the gene. Even for this purpose, we are using PCR. And if you want to study DNA polymorphism in the genome, using the known sequencing, you can use this polymerase chain reaction. So it has wide application in various fields. So genomic sequencing and study of DNA RNA as well as nucleic acid protein-protein interaction, you can use PCR. And also for analysis of AFLP, that is amplified fragment length polymorphism, you are using this PCR. So these are different amplification or the applications of PCR. So with this, I am concluding my first day workshop, and I am open for discussion and any questions. Yes, thank you very much. Yes, 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 tomorrow is the second day of my technical workshop. Thank you very much for the positive remarks. Thank you, Vivek, thank you very much. Thank you, Suresh, thank you, Vikas. Thank you, Abhishek, thank you very much. Thank you, guys.